important topic. So as the medical field continues to develop new medications or new diagnostic techniques, more prescriptions are being prescribed, and those pharmaceuticals, more pharmaceuticals are going to end up in aquatic systems. And current wastewater treatment doesn't target pharmaceuticals. Some of them are removed during wastewater treatment, but not because of targeted mint. They're just removed when everything else is removed. There's multiple sources, and I'm going to talk about a couple of sources. And there's currently no regulation or monitoring really of pharmaceuticals, so we don't really know if there's an abundance in one place or if a bunch gets leaked. We're not really keeping track of that. And there can be a wide variety of implications on ecosystems and organisms. And an increased awareness of this topic could potentially improve this and decrease pharmaceuticals being leaked into aquatic systems. So to research it, I wanted to look at where the pharmaceuticals are coming from and how it's affecting different organisms and how those effects might have a larger impact on the ecosystem, and if there's any possible solutions to this. So first I looked at a couple of USGS studies, and the first one was trying to determine where they're coming from. So they hypothesized that it might be coming from agricultural areas as livestock medication leaks into nearby streams, or wastewater treatment plants, and the main source would be humans there just from dumping unwanted pharmaceuticals or things like that. So they found that most detections were made downstream from wastewater treatment plants. So humans were the main source of pharmaceuticals, and there was very few detections from agricultural areas. And then the second study was they compared wastewater treatment plant effluent normal to wastewater treatment plant effluent that receives flow from pharmaceutical formulation facilities to determine if any of these pharmaceuticals are coming directly from those companies. And as you can see, there's a lot higher concentrations coming from pharmaceutical formulation facilities. So then I looked at how these pharmaceuticals play a role in the ecosystem affecting organisms. And this study was on European perch and damselfly larvae, so it was a predator-prey relationship. And they exposed them to varying concentrations of the psychiatric drug oxazepam. And they studied behavioral changes as well as accumulation of the drug in tissue. And an additional step that they took is they looked at whether feeding unexposed perch exposed damselfly larvae led to accumulation in tissue, so secondary exposure. Okay, behavioral changes before I go to the next slide. They saw an asymmetrical effect, so it did have an effect on the perch, but did not have an effect on the damselfly larvae, and it made the perch more bold. So as you can imagine, that could disrupt the food chain if the perch are more likely to go after the larvae. So then for the accumulation in tissue, this is the table that they had. So the first thing is this one right here, the unexposed fish eating exposed prey did have accumulation in tissue. So that's an important point that shows even if an organism isn't directly exposed to the pharmaceutical, it can lead to an effect. And then as expected, the exposed fish eating the exposed prey have the highest concentration of oxazepam. And then the damselfly have much lower accumulation of this in their tissue than the perch, which could lead to, or which could explain the behavioral changes in the perch, but not the damselfly. So this one, this study was similar to another talk that somebody gave earlier this semester. They studied concentrations of levonorgestrel, which is a common birth control, on the western clawed frog on its reproduction, and overall it had negative effects. So a decrease in gonadospermatic index, decreased oocytes, so just overall negative effects on their reproduction, which could also disrupt the food chain if this species population is declining. So after looking at a couple of animals, I wanted to look at how pharmaceuticals can affect algae. So they had varying concentrations of 4 different pharmaceuticals and exposed algae to them and measured growth rate, chlorophyll A, and lipid yield inhibition. When they exposed the algae to these pharmaceuticals, they observed cell swelling. That was just because the pharmaceutical disrupted their cell wall, leading to cell swelling. And in these charts here, as you can see, inhibition initially at low concentrations decreased, so it has had a positive effect on the algae. But then as concentrations increased, it had a negative effect and inhibition increased. So that positive effect you can see at low concentrations could actually be used as a possible solution to remove pharmaceuticals. So that's called phycoremediation, and I looked at this. They use microalgae, and it varies with algae species, but the study that I looked at used a native mixture of microalgae and took wastewater treatment effluent from a nearby facility and brought it in and measured pharmaceutical concentrations before and after exposure to the microalgae. And as you can see, the removal efficiency increased, and there was just overall less pharmaceuticals present after exposure to the algae. So you can see from the effect that it has on organisms that those could disrupt the food chain and have an overall negative effect on the ecosystem. So it's something important that we need to pay attention to. And phycoremediation is a possible solution to this. So although it would increase holding time at wastewater treatment plants, it is effective, not 100%, but it does help remove pharmaceuticals. And then I looked at a couple of studies on current threat to humans, and they determined that there's no current threat, but with increasing concentrations of pharmaceuticals, it could eventually become a threat. So it's just something that we should be aware of. Questions?
For your first study, can you back to that? Yeah, so what drug was this? What psychiatric drug was this? It was
Oxazepam.
What, do you know what that's used for? Or is,
Well, it is a PAM, which is similar to alprazolam, which is usually like an anxiety drug. So I'd assume it's something like that.
okay, so it made the perch less anxious and more bold, maybe?
Yeah.
Okay. That was the idea behind it. That's what they were sort of fishing for, so to speak.
I like it, Deb.
Okay, can you go to the next study? All right, well, did you get the general impression that, pharmaceuticals are more of a threat to vertebrates like fish and frogs, more so than invertebrates or algae,
Well,
based on concentration studies or
there's
things
a
they do?
lot more studies on vertebrates. So
Right.
I struggled to find papers for,
Right,
for
that makes
that,
sense.
but I feel like just based on what is available, it does have a larger impact on it.
Yeah, that makes sense to me, I guess, but I'm just curious if there were,
Right.
like, I don't know, mayflies are sensitive to a lot of things, but I'm just curious if they're even sensitive to birth control pills or things like that they get into.
Right.
So. Other questions? All right. Thanks, Bailey. And I agree with you.
I thought it was you're quietly, you forget.
Dev, while we have this brief pause for the exam, probably don't want us commenting on our own, correct?
No, for the exam, review 6 of the talks, but not your own.
Okay.
Unless you're going to give a really self-critical review, I wouldn't mind seeing that. Why am I talking this lame?
I just figure out a minute. I'm like, I should probably clear that up.
All right.
Okay, I'm Anna, And my topic is freshwater pollution, which is really broad. So I kind of broke it down into 3 branches of pollution that are a little more not regulated, either because they're newer or because the government just hasn't caught up to the science yet. So first of all, the importance of freshwater. We've gone over this multiple times in the semester, but the biggest thing is that we use it at home. It's not just irrigation, it's not just recreation. The average American uses 82 gallons of fresh water a day just in showering, doing dishes, washing your dog, whatever. So beyond that, why we should care about pollution in fresh water. So first of all, we have the overall water quality that we can detect with diatoms and mayflies. And you have to think about, if we run out of good quality streams, what happens to these creatures that are really important to the food chain? Where do they go? So that's something to keep in mind for sure. I also ran across a study that discussed the economic impact of freshwater pollution. And we, the taxpayers, all together spend like $4.3 billion a year to clean up like wastewater treatment plants and company contamination and what have you. So it has a huge economic impact on us, and it also can have a huge impact that can be irreversible on water fleas. So this study, published in the Evolution Letters, looked at the epigenome of water fleas, and they were trying to detect how sensitive this genome was to pollution. So they took water fleas and they put them some in clean stream water and some in stream water with pollutants in it. And then they looked at the changes in their DNA as far as the chemicals go. So their main marker was the methylation of cytosine. So you can see, the bottom row is the control group. So this is for cytosine, this is for glycine, I believe, and then this is for an enzyme, nucleoside phosphorylase. And then the top is the average increases and decreases they saw in the expression of those things. So it was a pretty significant study as far as seeing the effects just the pollutant can have on the DNA. And since it was affecting their DNA, they found that this was inherited in their future offspring. So they looked at, okay, we polluted up this water flea, it's showing this negative effect, let's take it out and put it in clean water, let's let it have kids in clean water, take those kids out, put it in more clean water, let it have clean water. And they found that even in the grandchildren, it still had this negative effect. It was directly inherited from grandchildren and so on. If one experienced pollutants, the rest of the generations generally experience pollutants, up to like 15 or more. So it's not something we can just fix later, is the takeaway. If it's happening in water fleas, it could also be happening in other ways with us and with our dogs and with other things we care about. So clearly I'm a dog person. That's all my examples today. So the first big unregulated branch is phosphate. It's kind of a necessary evil, because by the year 2050, we're going to have 10 billion people on the planet, and we can't feed everybody. So 80% of the food globally comes from farmers. And to put that more close to home, 50% of the food we get on a global basis comes within a 500-mile radius of St. Louis. That's where it's grown. So this is something in our backyard that we can impact through research and investigation. So basically, farmers use fertilizers with phosphate. It rains, that runs off into the streams, and you get this whole green mess that looks like a golf course, but it's supposed to be water. So it's not great. So our need to feed people actually has a negative impact on human health and wildlife. So some people are doing research on how to remedy this. So this Northwestern University study came up with what they call the PEARL membrane, which stands for Phosphate Elimination and Recovery Lightweight Membrane. I don't think it's a great acronym. I think they just wanted to use the name PEARL, but whatever, it works. So basically it's like a cellulose mat, and they covered it in manganese sulfide, and then they were able to adjust the pH and use this mat to sequester 99% of phosphate ions found in streams. So then, after all the phosphate was absorbed, they could actually recycle the phosphate while getting it out of the streams. And it works really well. So that's one way people are doing it. The second unregulated branch is microplastics, which we hear a lot about now, but there's not a lot of government guidelines set up for it because it's kind of new and it's just now catching up with us. So it's found everywhere. And generally, water treatment plants either don't have to or don't have the systems to filter the microplastics out of the water. So we're drinking them, they're getting into our bloodstream, it's a whole mess. And zooplankton also eat them and then they make their way up the food chain. They're really hard to get rid of. But generally we think of them in terms of the ocean because we always viewed microplastics in the rivers as rivers are a loading system. They're lightweight. It makes sense that they'll just flow through to the oceans and they won't linger. But there was a paper done, I believe it was published in the Proceedings of the National Academy of Sciences, and they looked at the hyperhyoid zone, pardon my French, and basically they found that an exchange occurs here at the headwaters where the surface water mixes with the groundwater and sediment underneath. So during this flip, it pulls the microplastics from the top of the surface water and traps them in the sediment below. So after they observed this, they then decided to chart the rate of how the microplastics travel now that they're in the stream bottom. And what they found was that it took about 5 hours for microplastics to travel 1 kilometer. But that was in high flow conditions. During low flow conditions, they tracked it, and it takes 7 years for microplastics to travel 1 kilometer. So they stay a long time, and headwaters are generally where a lot of our friendly organisms, like our water fleas, hang out. Very popular organism to study. And they found that when these organisms specifically ingest these microplastics, they become immobilized. They just kind of freeze in not an acute way, and then they get eaten, and then it throws off the whole food chain, and you have trophic cascade, and it just affects all of the stream health. So the last one we're going to talk about, which Blake talked a lot about already, is salt pollution. So thanks, Blake. It mainly comes from agriculture, mining, and apparently MoDOT. And it has a huge loss of zooplankton. It can cause algae proliferation, which is the green that we saw earlier. And I really investigated the government guidelines for this, for my topic of finding not very well regulated pollution. And I found that they give you kind of a range, and this is in the US, Canada, and throughout Europe. And they say, okay, you can have so much salt within this range. And even people who followed the lowest part of that range, the guidelines still exceeded what was good for the environment. So you still saw massive loss of biodiversity, even if you were well within that range. So this is something the government has also not kept up well with. So the 3 main takeaways I have for you are that pollution impacts can be irreversible. And if they're irreversible for water fleas, they could very well be irreversible for us. So that's something that needs more research. And some of the least regulated pollutants are phosphate, plastic, and salt. And then research is being done, but there's no quick fix. We're still trying to recover from things we did 80 years ago, let alone the stuff we're doing today. So bear that in mind. Here's some of my work cited. And are there any questions?
When you were looking at their last slide with the salt, and I would say you don't have to go through it, but when you were looking at the salt pollution, and I probably should ask this with his as well, is that some states use magnesium chloride instead of salt.
Yeah.
Did you see anything? Did they talk about those? I'm just more
The
curious.
papers I looked at were
Just at salt? Okay.
Yeah, they were pretty pessimistic.
It's still pretty bad. It's
No, I'm not saying it's good, but there are different effects from it.
It's very similar effects. It's not as harsh, but it still has the same effects, just not as like in lower concentration or in higher concentrations. It does the same as a lower concentration of normal salt.
Can you go back to your phosphorus study? So for phosphorus,
Oh, for
the
phosphorus,
sponge,
yes.
so, okay, yeah, so this Northwestern study, so this pearly sponges, you have sponges there, is this
Yeah,
from their study or did you put those in?
No, this one was in their paper.
Yeah, okay,
So this is what it looks like. I looked it up online. It looks like just like a foam mat. It's like
Yeah.
black. And yeah, they say it's constructed with cellulose and then they like dip it in manganese sulfide.
right.
And that allows them to like mess with the pH.
And would the idea be that would you put this in like a wastewater treatment plant or would you put it
Yeah,
alongside?
they put it in a lot of places. Some of them They put it just like in a eutrophic lake or
Really?
stream.
Yeah.
And then some of them they put in wastewater treatment plants. And they saw similar results in both. The wastewater treatment had to have like more because it's
Right.
gross. But
Yeah, okay. Other questions? All right, thanks. It's all clap. All right,
For my topic, I chose the effects of copper pollution on freshwater fish. And we talked about it. So, copper is really prevalent, in wastewater, and other sources. A lot of urban runoff has copper from brake pads, in cars. And it's really, really widespread, and so that's why I want to investigate it. And so one of the biggest effects of copper is that the fish will decrease their activity, or one of the natural behaviors is that they will decrease their activity when they sense a predator. And it could be pheromones from the predator, like an actual like scent essentially from the predator, or if the predator is to injure a fish near them, they can also smell that and then swim away or freeze. And so they use their olfactory senses for that. And the copper has been shown to decrease olfaction. So they, as I said, they have less detection of pheromones and a lot of cases from other prey, but that also affects their breeding and their reproduction. Because if they can't sense other fish and like if they can't return to their streams, especially in salmon, a lot of their olfactory senses go into where they return to their original streams where they were spawned. If they can't find that stream, then they can't really do that. Copper nanoparticles have been shown to alter the radio, reduce and oxidize the ion, which damages the DNA and cell walls and just kind of blows everything up. So the prey movement, over here you see the initial, these are like the unexposed and then exposed, but they're normal relatively. It would go, like they would stop moving. So you see how whatever, there's no predator, and then they move down once they sense the predator, and then they stop moving almost altogether once they sense a water substance, in addition to but with copper, it goes down a little bit, but almost not at all. And then, as the copper concentration increases, they notice a very, very large increase in the swimming speed, which means that they were moving a lot more and they were a lot easier to sense. Also noticed that the predators were, because they were able to detect them a lot easier, they were able to successfully capture them a lot faster. So success was defined by them capturing them on the first attempt. And so at zero concentration, it's like pretty low chances, but as you go higher, it is very, very high, like 30% capture success rate on the first chance. And as you can see over here, the control group survived 75% of the time, where the exposed group survived 25% of the time when in presence of a predator. And they theorized that that was because the olfactory senses were decreased. And then you can see over in this graph over here the different concentrations of copper per liter, and then the control group and their survival times. And granted, they die eventually, but as you go up in concentration, you can see there's a decrease in survival time and the portion of the survival and the midpoints on all of these graphs were shifted to lower. So I then wanted to investigate the difference between the nanoparticles of copper and copper sulfate in the waterways. And so different effects on olfaction. The copper sulfate decreased the responses, but they didn't completely eliminate them, whereas the copper nanoparticles did completely eliminate those like behavioral responses where they would freeze up. And then I also found that the copper sulfate damaged cilia in the olfactory rosette, which is just like in the noses, whereas the nanoparticles caused the oxidative stress. So it was a different mechanism to that damage. And you can see over here, this is, they measured the amount of time they crossed the line in the tank to see their like movement, because otherwise you couldn't tell if the fish is moving or not. And you can see that the control groups were like not moving as much in the sense, in the presence of predator, and then the non-control groups were moving a lot more. And you can also see over here the time inactive. The control groups took a lot longer to resume activity after being presented with that information, whereas the experimental groups did not. And you can see that the nanoparticles had less of an effect than the salt mitigated nanoparticles. And this is to demonstrate the oxidative stress that the nanoparticles would cause. And so this is the total remain, and this is the reduced and the oxidized. And so you can see that the ratios over here, the oxidized and the reduced, are changing. And so you can see that the nanoparticles pretty drastically decrease that ratio, which is important to keeping the cell and the DNA from being damaged.
For your studies, what were the taxa fish that were used?
This is the spine loach. This is the rainbow trout, and this is the boas.
Like for the first study, what was that done on?
This one was on the spine loach.
Okay, and it was getting eaten by some bigger fish.
I believe it was cutthroat trout.
Yeah, that makes sense.
And then this one was the rainbow trout up here, and this one was the boas.
Okay. Wait, is there a difference in terms of where do the copper nanoparticles come from? Is that mainly an industrial thing, or is it just?
It's mainly an industrial thing because the way that the copper would enter like the waterways, if it was copper sulfate, mostly like runoff or agricultural products.
Okay, yeah. So what do we use? I mean, is someone making copper nanoparticles? I know nanoparticles are a big industry.
It's more just a byproduct of other industries.
Yeah, okay. All right. Other comments? Questions? Okay, thanks, Mike. All right, that's all for today, then. So we'll have a busier time on Tuesday. I think, we did 3 talks in half an hour, so we might be fine.